Bismillahirrahmanirrahim. Assalamu alaikum, dear students. Hope you and your family in the best of health. Dear students, this lecture is for grade 10, subject chemistry, uh, chapter number 12, exercise, and we will also start chapter number 13. This is my 22nd lecture, and I am your lecturer, Mrs. Saira Jamal. We will do question number 13 and 17 from Think Tank according to the reduced syllabus pursued by the Federal Board. The question number 13 is identify A, B, C, D in the following. The first one is propene. After bromination is converted into A, and after reaction with alcoholic potassium hydroxide, A will be converted into B. So what is A and B? We have to identify A and B. Propene, prop for 3 carbon atoms. Propene after bromination, which is also called halogenation or addition of halogen or addition of bromine. One bromine will attach with one carbon atom and the second bromine atom with the second carbon atom. So it will give us propane because all the carbon-carbon bonds are now single bonds. So this is called one, this carbon is called carbon number 1, 2. 1, 2, there are two bromine atoms, dibromopropane. Name of A is 1,2-dibromopropane, and this dibromopropane, which is formed in the first case, is dibromopropane after alcoholic reaction with alcoholic potassium hydroxide. They will give us, what will they give us? They will give us one propene. They will give us one propene. This alcoholic potassium hydroxide, you can see in this first case, which is 1,2-dibromopropane. So two bromine atoms are present. One bromine is present at carbon number 1 and one bromine is present at carbon number 2. So if you remember, I told you that bromine from carbon number 1 and hydrogen from carbon number 2 will eliminate out by the potassium hydroxide. And then in the second step, the second bromine and hydrogen from the first will eliminate out by this potassium hydroxide. So for this purpose, we need two potassium hydroxide molecules to give two potassium bromide molecules and two water molecules. So the final product which is formed is one propene because this is carbon number 1 and double bond is present at carbon number 1, so it is called one propene. So what is A? A is bromination of propene will give us 1,2-dibromopropane, and then this alcoholic potassium hydroxide when react with this 1,2-dibromopropane, dehydrohalogenation will take place. Dehydrohalogenation means hydrogen from one carbon atom and halogen from second carbon atom will eliminate out with this potassium hydroxide to form potassium bromide and water, and the final product we will get the propene again as a final product, that is B is one propene. A is one propene, B, the reactant is propene, and the product also is one propene. B part, ethyl alcohol when react with concentrated sulfuric acid, it will give C, and C after bromination will give D. So this ethyl alcohol and concentrated sulfuric acid high temperature indicated that this is actually a dehydrating reaction because concentrated sulfuric acid we use as a dehydrating agent. So this dehydrating agent, hydroxyl group from one carbon atom and hydrogen from the second carbon atom will evolve out in the form of water. As a result, initially there are 3 hydrogen atoms, but at the product side 2 hydrogen are left. And this hydrogen also will evolve with this hydrogen and will form water. So the product C is ethene. Name of product is ethene. This ethene after bromination or addition of halogen, one bromine at one carbon atom and second bromine at the second carbon atom will give us 1,2-dibromoethane because the bond between carbon is single bond. So it will give us 1,2-dibromoethane. Now question number 17 is we have to differentiate between ethene and ethine. So there are two important tests for the differentiation of ethene and ethine. The first test is ammonical silver nitrate test. This is called Tollens test. It is also called silver nitrate test or Tollens test because we are using a special type of reagent which is called Tollens reagent. So alkyne give a white precipitate with the Tollens reagent, but alkene does not form any type of precipitate. So formation of precipitate with this Tollens reagent, that will be silver acetylide, ammonia and water. So these silver acetylide actually give us white precipitate, but ethene will not react with Tollens reagent, so no reaction. So this is one of the identification of ethyl. Second test is ammonical cuprous chloride test. With an ammonical cuprous chloride, alkyne give red precipitate while alkene does not give any result. So ethyl when react with ammonical cuprous chloride, it will form red color precipitate of copper acetylide, ammonia and water. And ethene they does not react with this ammonical cuprous chloride. No reaction takes. So these are the two important tests which we can use to identify or differentiate ethyne from ethene. Now we are going to start the next chapter, that is chapter number 13, that is about biochemistry. What is biochemistry? The study of the chemical substances and vital processes occurring in living organisms. Because bios means life and chemistry means changes that takes place in living organisms. So the study of chemical substances and vital processes occurring in living organism, biological chemistry, physiological chemistry, the chemical composition of a particular living system or biological substances through biochemistry. In simple word, what is biochemistry? Bios mean life and chemistry. I told the changes which takes place in living organism. So all those changes which takes place in living organism we study in a branch of chemistry called biochemistry. The first topic of the biochemistry is the important nutrients. You know that many important nutrients are present in our food. Those important nutrients are carbohydrates, proteins, lipids, and vitamins and nucleic acid. All these are important nutrients which are required by our body. One of the important nutrients is carbohydrates. Carbohydrates are the most abundant class of organic compounds. Carbohydrates are also called hydrates of carbon. They are also called hydrates of carbon. General formula for carbohydrates are CxH2O whole Y. They are Y moles or Y number of water molecule with carbon atoms. Plants synthesize carbohydrate through a process called photosynthesis. Plants convert carbohydrate into starch and cellulose, while animals convert carbohydrates into glycogen. Carbohydrates are monomers and polymers. Monomers mean they are made up of single unit, and many single units combine together to form polymer. Poly means many and merge means parts. Mono means single and merge means part. They are made up of single part. They are made up of more than one part. Of aldehydes and ketones that have numerous hydroxyl groups attached. All aldehydes and ketones have many hydroxyl groups means OH groups. As I told you that carbohydrates, they contain aldehyde or ketonic functional group. Aldehyde functional group, you know that this is carbonyl carbon atom which has attached at one side hydrogen is attached and at other side any alkyl group may attach. But in case of ketone with the carbonyl carbon atom at both sides two alkyl groups will attach. As I told you that the plants they synthesize food by a process called photosynthesis, and photosynthesis for photosynthesis the required reactants are carbon dioxide, water, sunlight, definitely chlorophyll, and it will give us glucose and oxygen. Glucose we use for energy purposes, and oxygen we use for respiration. So these are the two products which are given by photosynthesis. Carbon dioxide react with water in the presence of sunlight, and chlorophyll to form glucose, sugar, or and oxygen. Classification of carbohydrates. Carbohydrates are classified as monosaccharides or oligosaccharides, polysaccharides. Monosaccharides have the monomers or simple sugar units from they have sugar units like only you can say they are composed of one sugar unit, and oligosaccharides are composed of 2 to 10 sugar units, and polysaccharides are composed of more than 10 sugar units. Monosaccharides are the simplest carbohydrates. They are white crystalline solid. They are soluble in water and they have sweet taste. They cannot be hydrolyzed. Hydrolyzed means by the breakdown of a molecule by the addition of water. Water molecule itself will also breaks or split up and also will split the monosaccharides or carbohydrates. So monosaccharides, they cannot be hydrolyzed because they are already in their simplest or hydrolyzed form. They are reducing in nature. Reducing in nature means that they loses or they can give hydrogen atom. So by losing hydrogen atom or by gaining hydrogen atom, they by themselves get oxidized but reduces the other substances. That's why we call them, they are reducing in nature. So all monosaccharides, they are reducing in nature because they contain OH group at carbon number 1, and they can give that hydrogen atom so they can be easily oxidized and reduces the other substances. Monosaccharide molecules can rotate the plane polarized light to right, clockwise, or anti-clockwise. So they are called dextrorotatory or dextrose sugar. If they rotate the plane polarized light at right side, then they are called dextrorotatory or also called dextrose sugar. In this image, you can see that we have an ordinary light which spread in all direction. But when this light passes through polarizer, the polarize this light in one direction. So you can see the direction of light is only single direction. And when this single direction light is passed through another slit, you can see they are rotating in two directions, clockwise or anti-clockwise. The clockwise direction is called dextrorotatory, and the anti-clockwise direction is called levorotatory. And the instrument which is used for this purpose is called polarimeter. This is the image of polarimeter. In this polarimeter, this process takes place. Ordinary light enter, and then it is polarized by passing it into the polarized, and then after polarizer, it passes through the slit, and then that slit you can see through the eyepiece that you can see through the eyepiece that the direction of this light is either clockwise or anti-clockwise. If it is clockwise, it is called dextrorotatory. If the rotation is anti-clockwise, then it is called levorotatory compound. They have general formula CH2O whole N, where N is 3 to 6 carbon atoms. Monosaccharide have the carbon atom from 3 to 6, and they have general formula CH2O whole N. They are further classified on the basis of a number of carbon atoms. If the monosaccharide contains 3 carbon atoms, minimum is 3 carbon atoms. There is no carbohydrate which contains carbon atom less than 3. The smallest, you can see the simplest one is 3 carbon containing monosaccharide, which is called trioses. Tri means 3, okay? Trioses. The 4 carbon containing carbohydrate or monosaccharide is called tetroses. Then pentose is 5 carbon atom, and hexoses have 6 carbon atoms. So these monosaccharides are classified on the basis of number of carbon atoms present in them. So trioses, tri means 3, tetra means 4, penta means 5, and hexa means 6. So trioses 3 carbon atoms, tetroses have 4 carbon atoms, pentoses have 5 carbon atoms, and hexoses have 6 carbon atoms. Examples of monosaccharide. The most familiar monosaccharides are glucose and fructose. Glucose and fructose, they have same molecular formula like C6H12O6, and they are hexoses because they contain 6 carbon atoms. But glucose is called pentahydroxyaldehyde, and fructose is called pentahydroxyketone. The most familiar example are glucose and fructose. They are simple sugar. They cannot be hydrolyzed, and they are pentahydroxyaldehyde or pentahydroxyketone, because glucose is an aldehyde and fructose is a ketone. And pentahydroxy means what? Penta means 5 and hydroxy means contain hydroxyl group. So all carbohydrates, if they are trioses, tetroses, pentoses or hexoses, all contain hydroxyl group in them. Although the hydroxyl groups are different in different species or in different compounds, but glucose and fructose are the most common and most familiar examples of hexoses because their formula is C6H12O6. So C6 shows that they have 6 carbon atoms. So means that monosaccharides, they are classified on the basis of number of carbon atoms and how many carbon atoms are present in them. So glucose is called pentahydroxyaldehyde, while fructose is called pentahydroxyketone. These are the images of glucose and fructose. You can see the carbon number. There are 1, 2, 3, 4, 5, 6. 6 carbon atoms are present in glucose. These structures are also called open chain structures of glucose and fructose. We also have closed chain structures of glucose and fructose. And here again, 1, 2, 3, 4, 5, 6. 6 carbon atoms are also present in fructose. It means the carbon numbers are same. Coming towards the functional group, you can see that this whole group is R, then C double bond O, C double bond O and H. So R, C double bond O, H is which functional group? It is a functional group called aldehyde functional group. Aldehyde functional group is the functional group where the carbonyl carbon is attached at one side with alkyl group, R group, and at one side is attached with hydrogen atom. So glucose is an aldehyde. One more thing is that they are also called pentahydroxy aldehyde. So aldehyde is confirmed because they have aldehydic functional group. In this case, ketone is confirmed because it contains ketonic functional group. Because carbonyl group is in the middle, this group is called R group, and this is also called R group. So two R groups are attached at the two sides of the carbonyl carbon atom. That's why they are called ketones and aldehydes. As I told that they are also called pentahydroxy because they have 5 hydroxyl groups. Let us count: 1, 2, 3, 4, 5. So you can see there are 5 hydroxyl groups are present in glucose, and those 5 hydroxyl groups can be replaced by any other group. So this is called pentahydroxy and aldehyde. In case of the ketone, as ketonic functional group is present, and number of hydroxyl groups are 1, 2, 3, 4, 5. So as 5 hydroxyl groups are present in fructose as well, so this is also called pentahydroxy. But as ketonic functional group is present, so we will call it pentahydroxy ketone. And glucose is called pentahydroxy or hydroxyl aldehyde. Now, what is the another difference between the two? This glucose and fructose is that the main difference is how to draw the structure of glucose and fructose. You can see, first of all, you have to make a 6-chain carbon atom: 1, 2, 3, 4, 5, 6 for glucose, and 1, 2, 3, 4, 5, 6 for fructose. Then draw the carbonyl carbon atom. Carbonyl carbon atom we have to draw at carbon number 1 for glucose because it is an aldehyde. While for ketone, the hydroxyl group will be present, you can see above as well as below the plane. But we have to draw first the long chain carbon atoms. Now, as I told you that, or if you see the structure of glucose, all the OH groups are present at right side except carbon number 3. At carbon number 3, OH is present at the left side. Okay, all the rest of hydroxyl groups are present at the right side except carbon number 3 where hydroxyl group is present at the left hand side. And same is the case in fructose. All the hydroxyl groups are present at the right hand side except at carbon number 3. The hydroxyl group is present at carbon number 3. The rest of the structures are same and the functional groups are different. It is carbonyl having at one side alkyl group and at one side hydrogen is attached. So in this case, at one side R group and the second side also two R groups are attached at each side of this glucose and fructose. So, dear students, this was all about the carbohydrates, its classification, and about some of the examples and some of the properties of the monosaccharide. I told you that all the monosaccharides, they are sweet in taste, they are crystalline solid, they are soluble in water, they are reduced to sugars, they can rotate the plane polarized light in clockwise direction, they are called dextrorotatory or dextrose sugar, and the instrument which is used for this purpose is called polarimeter. Carbohydrate are classified as monosaccharide, oligosaccharide, polysaccharide. This classification is on the basis of sugar unit present in each. Monosaccharide has 1 sugar unit, oligosaccharide has 2 to 10 sugar units, and polysaccharides have more than 10 sugar units. Then monosaccharides and their properties. Monosaccharides are then classified into trioses, tetroses, pentoses, and hexoses. And example of hexoses are, I told you, are the glucose and fructose. Glucose is an example of pentahydroxyaldehyde because it has aldehydic functional group, and fructose has a ketonic functional group. And about the plane polarized light, I told you that a polarimeter is used for this purpose, and the ordinary light which move in all the direction when it passes to the polarizer, the direction of light is in one plane, and when this light passes through the specimen, that may be D glucose or L glucose, D glucose will rotate the plane polarized light towards clockwise direction, and the L glucose will rotate the plane polarized light in anti-clockwise direction. Then by this way we will get the D glucose and L glucose. So hope you understand this lecture. Inshallah, we will do the other classes of the carbohydrate like oligosaccharide and polysaccharide inshallah in our next lecture. Till then, take care and